Good evening and welcome to the McMaster Center for Climate Change public lecture on COVID-19, Seasonality, Transmission, and Protection. My name is Altaf Farin. I have the privilege of serving as Director for the Center for Climate Change and hosting tonight's talk. We have done many of these talks over the years. However, this is our first online talk. And this is also our largest talk with more than 600 attendees. We are recording this talk so that you can share it with family, friends, and colleagues. Tonight, we will be hearing from infectious disease specialist, Dr. Mark Loeb. After Dr. Loeb's talk, you will have the opportunity to ask questions. Please type your questions in QA box on your screen. Some of you have already submitted questions in advance. We'll try to cover as many as we could. To introduce Dr. Mark Loeb, I would like to invite the Dean for the Faculty of Science, Dr. Maureen McDonald. Maureen is first woman to serve as Dean for the Faculty of Science. Our faculty is the largest at McMaster with nearly 8,000 undergraduate and more than 600 graduate students. In addition to leading 200 faculty and 100 staff members, Maureen also runs a research lab in the Department of Kinesiology and has been nationally recognized for her excellence in student mentorship. Maureen.
Thank you so much, Altaf, and good evening, everyone. While we have an international audience watching tonight's lecture, I would like to start by acknowledging that McMaster University is located on the traditional territories of the Mississauga and Haudenosaunee Nations, and within the lands protected by the Dish with One Spoon Wampum Agreement. Thank you for joining us, no matter where you're joining us from, for this very important and timely lecture as confirmed COVID cases surge in our province. Fortunately, we have had only a few confirmed cases at McMaster University. We were among the first universities in Canada to move to remote teaching and learning and to close our residences for the current academic year. Before introducing our keynote speaker, I'd like to acknowledge Altaf's excellence as a researcher and as the founding director of McMaster's Center for Climate Change. Altaf is as accomplished as he is modest. He is among our most widely cited researchers at McMaster. Altaf established the Turkey Point Observatory, which examines the impacts of climate change and extreme weather events on forest ecosystems and their restoration in the Great Lakes regions. He also works on the development of hydrologic and climate models. Our Center for Climate Change, which is our host for this evening, is one of 5 research-intensive centers and institutes in the Faculty of Science, and among the more than 70 centers and institutes across McMaster University. Our centers and institutes tackle complex, multidimensional challenges by bringing together multidisciplinary teams of faculty and student researchers. Our teams then collaborate with industry, government, and academic partners. And we see this very prominently during the current pandemic, where our institutes and centers have been taking front stage with tackling many important questions. Our Center for Climate Change is advancing discoveries and knowledge about the impacts of climate change on the environment, ecosystems, water resources, and human health. Community outreach and engagement is a priority for all of our centers and institutes. It's why our Center for Climate Change is hosting tonight's lecture in collaboration with Global Water Futures and the United National University. There may be no challenge more complex than climate change. Many of the lessons we are learning on how to mobilize and tackle a global pandemic can also be applied to addressing global climate change. As winter and flu season approaches here in Canada, we wanted to hear from another internationally renowned researcher. So we are very excited and grateful to have McMaster University colleague, Dr. Mark Loeb, join us this evening. Dr. Mark Loeb is a medical microbiologist and infectious disease physician. Dr. Loeb is also a McMaster University professor in the departments of pathology and molecular medicine, as well as health research methods, evidence, and impact. Dr. Loeb is the Division Director of Infectious Diseases and Head of Service for Infectious Diseases at McMaster, and holds the Michael G. DeGroot Chair in Infectious Diseases. Dr. Loeb will also be a key contributor to McMaster's new Global Nexus for Pandemics and Biological Threats. Dr. Loeb has served on 45 international, national, and provincial advisory committees and published 342 peer-reviewed papers and 28 book chapters. I'm really glad that I'm not going to read all those titles for you this evening. Dr. Loeb has been recognized with a gold medal in medicine from the Royal College of Physicians and Surgeons of Canada, an America Investigator Award from the Society of Healthcare Epidemiology of America, and the Jonas Salk Award from the March of Dimes. Last month, Dr. Loeb was announced as a newly elected Fellow of the Royal Society of Canada, one of the highest honors possible for a scientist and physician in Canada. Dr. Loeb led cluster randomized trials to establish herd immunity using influenza vaccines in Hutterite colonies, along with randomized trial of surgical versus medical masks for influenza. He is now conducting a similar randomized trial in healthcare workers on COVID, along with a cohort study of COVID-19 in Hutterite colonies. So on behalf of McMaster's Center for Climate Change and our Faculty of Science and our other partners this evening, we are thrilled to have Dr. Mark Loeb join us this evening. Dr. Loeb?
Thank you very much. Thank you very much, Maureen, for the kind invitation, and thank you, Alta, for inviting me. So I'm just going to get these slides set up here. So what I'm going to be speaking about is the COVID-19 in terms of seasonality, transmission, and protection. And what I'd like to do is first start off with just a brief introduction simply about the virus and about the immune response, just to sort of set the stage. And then I'll go through a number of questions. And the first question is, how is COVID-19 or SARS-CoV-2 transmitted? And this is it's a very controversial question. Some of you might be aware that the CDC has sort of been going back and forth about communicating whether COVID-19 is aerosol transmitted or not, or is it just droplets? So I'd like to get into that controversy. From then, I'd like to move on to seasonality and specifically focus on the effect of cold weather and humidity on the effect of the spread of the virus. And obviously that is important as we go into a season with those conditions. And then finally, I'd like to talk about, on an individual basis, how we can prevent transmission of COVID-19. So to start off, this is a slide from the World Health Organization as of last Friday, looking at the enormous burden of COVID-19. Over close to 34 million cases worldwide and over 1 million deaths. So an enormous toll that's been taken with this pandemic, and it doesn't look like it's abating. So it's going to be with us, unfortunately, for some time. This slide is of the virus, the COVID-19 agent, which is the SARS-CoV-2. So it's an RNA virus, and I'd like to just bring your attention to this little piece here called the spike glycoprotein. So spike glycoprotein is what the virus will attach to when it's what it uses to attach to a cell. Okay, so and it's important to know about because this is the antigen that vaccines are using to produce an immune response. So for the most part, most vaccines are using the whole part of this glycoprotein, and there are some vaccines that are just looking at using this receptor binding domain. This slide is just to illustrate that when a virus infects a cell, what it does is it hijacks the protein-making machinery of the cell. So a SARS-CoV virus, like any virus, will enter the cell, and then it will replicate itself. And it's important to know that the antiviral drugs, and these are drugs that are that work well at the very beginning of the infection, not later on, will target different pathways within this. So some of the President of the United States was treated with a wide range of cocktail of medications that included some antiviral drugs. So what happens in a successful infection is that the virus will replicate itself, and then it will leave the cell and get these virions that will go and infect other cells. So what happens is, as someone is infected with the virus, the cell will replicate as I've shown you, and then it'll create a pathology. And this happens maybe 5 to 7 days into the illness, sometimes a little short, a little less longer, but it's the inflammation that occurs in the lungs. So you can see here that there are a lot of inflammatory cells, and this is where steroids or dexamethasone help dampen the response. So the antivirals are effective early on during the infection, and steroids are effective later on. So it's really important to understand also the immune response to the virus. And this is an interesting study where they looked at, it wasn't very large, they looked at 25 or 30 individuals, and they were people who got just moderately ill. They didn't get so sick. And what the investigators did is they looked at the adaptive immune response. So that's the part of the immune system that has a memory. And they looked at CD4 cells, those are T helper cells, and CD8 cells, and those are killer T cells. And they found that 100% of those people who were infected mounted a very effective or very robust CD4 response. So that's good. And 70% had a CD8 response, and the T cell response correlated to the ability of the body to produce antibodies, so IgG and IgA. So these are potential correlates of protection. So we don't know exactly what will correlate with protection until we follow people, until there's vaccine trials, but it's possible that it could be either a neutralizing antibody or a T cell sort of response. Now, what's really interesting in this study is they didn't only look at people who were known to be infected with COVID-19. They looked at a number of people who had no infection, okay? And they looked at their response to COVID-19, and they found that 50% of them had a T helper cell response, and 20% of them had a CD8 killer T cell response. So that's really interesting, because it means that even if you've never, been infected with COVID-19, if you've been infected with a seasonal coronavirus, you might mount a response. And it's just hypothetical, but it really is a question as to whether sometimes young, otherwise healthy people get really ill and sometimes die of COVID-19. And you wonder whether it's because of perhaps a lack of exposure to these previous viruses, common cold, coronaviruses, or they perhaps weren't able to mount an immune response, so there was no cross protection. That's one possibility. The other possibility is they were just exposed to a very high load of the virus, and alternatively had some sort of rare immunological polymorphism that led to them getting very, very ill with infection with COVID-19. Okay, so that's just a bit of background. Now, what I'd like to get into is the question. So the first question, again, it's super controversial, is how is COVID-19 transmitted? And this slide is just a very simple diagram that illustrates all the possible paths for transmission. So one of them is a large droplet. So that's sort of a spray sort of transmission, and that's when someone almost coughs or spits in your face, that there'd be really large particles that would infect the mucosa of the pharynx or your nose, something like that. So that's the droplet spread. The aerosol spread occurs when someone perhaps talks or coughs close to you and you actually breathe in those aerosols. So those are smaller particles and that they can make their way deep into the lungs to cause illness. So this is part of the debate. How much of this transmission is this large droplet? How much is aerosol? And it's very nuanced, and we'll talk about that some more in a few minutes. The other possibility is just direct contact. So if you're shaking hands with someone, for example, that's another form of transmission. And the other related to direct contact is if you're just contacting the environment. It's called fomite transmission. And there's really less evidence for fomite transmission. It really seems to be more of a large droplet aerosol type of transmission that's most important. So it's interesting, part of this controversy is this traditional dichotomy. So if you go to the World Health Organization website or old CDC sites, they always refer to either droplet or airborne spread. And they use this dichotomy of 5 microns, which is pretty small. And they say, look, if a particle is greater than 5 microns, this is large droplet, and it'll fall to the ground within 2 meters, and that's it. If the droplet is much smaller, if the particle is much smaller, less than 5 microns, well, then it'll get suspended, it'll just float around in the air, and it'll travel for long distances. So traditionally, most respiratory viruses like the flu or adenovirus, RSV, all of those, we usually think of those as droplet, and we think of things like TB, tuberculosis, which is a bacteria, not a virus, but measles or chickenpox, we think of those as airborne. But more and more aerosol biologists are telling us that based on the interpretation, first of all, they don't even know where this dichotomy of 5 microns really came from, for one thing. And the other is there's older experiments and there's newer ones that suggest that it's not just one, it's not either a really large particle or a really small one, but it's a continuum, so that even at short distances, you could be infected with droplets, but you could also be infected with aerosol particles or airborne particles at short distances. So I think maybe we have to sort of make a disconnection with this cluster of droplet characteristics versus airborne characteristics. So the idea basically is that some droplets might be larger, they evaporate over time, they float around, or sometimes, depending on the environmental conditions, they just might drop. So it is basically a continuum. But what's really important are the inferences. Well, what does that mean to protection? And I'll talk about that in a few minutes. So as I was mentioning, the size of the particle is really important. It depends on how long it stays aloft. So very small particles will stay aloft and travel further. Large particles will fall to the surface. And, I mentioned this dichotomy of 5 microns, and aerosol biologists are telling us that that's probably, it's really tenfold. It's probably 50 or 60 microns is a cutoff if there is one. But the size also is also important in terms of where the particle will deposit in the respiratory system. So very small particles are more likely to make it all the way down to your lungs, to the extreme, to the alveoli. And it also is really important, and there's a lot of controversy around this, about how efficiently it's removed by mask, or what kind of face mask you have to wear. And I'll be talking about that in a few minutes as well. So this is just to illustrate transmission by path of exposure. So large droplets are sprayed into the body. The aerosols you breathe in, and depending on their size, they could go all the way down into your lungs. And illustrating this in another way, the spray-borne, those large ballistic droplets, they just end up around your throat, and that's about it. The ones you inhale are smaller, and they could go through they might be less than 100 microns. It's called nasopharyngeal. They're coming down here. Thoracic, 10 to 15 microns, going midway, and then the ones that really you inhale and they go all the way into the lungs. So that is summarizing the importance of the size of these particles. Now, people have looked at the aerosol stability. So basically the question is, how long does the SARS-CoV-2 or COVID-19, how long does it really hang around in the air? And this was a letter to the New England Journal of Medicine, where the investigators looked both at SARS-CoV-1 and SARS-CoV-2 in red. So you can see here that it's really staying stable in the environment as an aerosol for several hours. And people have done more complex studies looking at the aerodynamic analysis of SARS-CoV-2. This is a study from China, where basically they went to several hospitals and they took this big filter and they just started sampling the air, started sampling the environment to see what they could find. Now, they weren't looking for viable virus, so they weren't looking for virus that could go in and that could biologically necessarily infect someone. They were looking for pieces of nucleic acids. It's almost like a footprint of the virus. And they found relatively high concentrations in areas where people were, healthcare workers were removing their protective gear. They found that in 2 hospitals, and they found some concentration of COVID-19 in the medical staff office. So this is where, these types of studies is where there's sometimes a discrepancy between inference. So sometimes people who are more doing, experiments, aerosol biologists will say, aha, yes, this is proof that there's aerosolization and that's causing transmission of infection. Whereas the clinicians, the epidemiologists who work in hospitals might say, hey, wait a second, that's showing that there's RNA all over the place, but you haven't proven that the virus is actually being transmitted this way. So often a lot of this controversy is people looking at the same data and interpreting it completely different, and that's really a running theme. This is more of a medical view, so this was from a medical journal as compared to an aerosol biology journal, where the authors here were making the argument that it's more of a droplet spread. So they said they looked at the reproductive number, that's the number of secondary cases that occur from an index case, saying, well, if it was measles, it would be 5 or 6. This is 2 or 3. That's more like droplet, things that we're familiar with, like the flu. And that the secondary attack rate, the number of other people who are infected from one person, is relatively low. There's high risk for household members that are likely to be really close together. All of this information they synthesized and said, well, we really think this really looks like droplet. And with healthcare workers wearing surgical masks, the infection rate was relatively low. So it's all part of that argument. But I think ultimately one has to say there are multiple transmission routes, and it seems as though most of the transmission is really at short distance. So whether we call that droplet or whether we say, oh, well, there might be smaller aerosols, but it's transmitted at a short distance. I think to me, that's an important take-home message. And then we see these types of scenarios. This is the famous, outbreak following a choir practice in Washington State. This is a slide from the MMWR from the CDC. So here you had a choir practicing once, and then someone who was sick, and there was another choir practice, and you see this sort of point source outbreak, sort of like a crescendo, decrescendo, suggesting that people were getting sick in a very short period of time. So some people look at this and say, this is a so-called super spreading event. And they make the inference to say, well, this is definitely aerosolized. They were singing, and there's experimental evidence that there could be aerosols, during singing, coughing, even talking. Whereas other people sort of looked at their activities a little more carefully and said, well, you know, these people were intermingling quite a lot. They go for coffee breaks and share snacks or whatever. So it's not totally clear that this is, you know, was an aerosol type of event. And just to finish that thought, I just want to say that I really think that some of the debate, sometimes it's a little bit too antagonistic, and I think we have to just you know, agree sometimes to disagree, that we don't always know, and that you can never say it's transmitted, you know, 100% one way or 100% the other way. Personally, I'm convinced that there are some events that were transmitted by aerosol. For example, there was this outbreak in a nursing home in Montreal where the ventilator system failed, and the attack rate, that is the infection rate, was like, oh, something like 95%. Everybody was infected. That to me is pretty convincing that that was aerosol transmission. So, okay, so another sort of somewhat of a controversy, I'd say, is the role of children. So there have been a number of studies, and there's sort of been a conventional wisdom that, well, you know, COVID-19, children don't really get infected, and they don't really get too sick, you know, from it. And it's true, I'd say it's true that from the data that exists, that the likelihood of a child getting really ill from COVID is not the same as adults or individuals who have comorbid conditions. But to me, the sort of more controversial thing is the reports are that they're less likely to be infected or less likely to transmit it. And part of the problem is that a lot of this data is based on public health reporting, contact tracing, where there's an outbreak and people go talk to parents or whatever. You can't really understand it very well unless you're doing, you know, a lot of swabs, a lot of very close follow-up. And this is what we want to do in this cohort study. So we've been doing studies among the Hutterites. Hutterites they're Anabaptists, sort of like the Mennonites. They live in Alberta, Saskatchewan, and Manitoba. And we've been doing studies on influenza vaccine in these colonies for, you know, close to 15 years. But what we want to do now is just very carefully follow entire communities, entire colonies of 80 to 120 people forward in time, do swabs, look at baseline antibodies. You know, I showed you that slide of the antibodies before, just to see, is this a correlate of protection? And because the colonies, the population is very young, 25 to 30% of a colony is entirely made up of children, I think that, you know, if there is transmission of COVID-19 from children, we will definitely find it in this community. It's been very unfortunate because this community has been very unfairly stigmatized because of unfortunately publicized outbreaks in their community. So we're trying to work very closely with them to do this study. Okay, so moving on to the second question, which was, how might cold weather and humidity affect the spread of COVID-19? So this slide just summarizes the relationship between humidity and aerosol size. So you could see that as there's humidity decreases, the aerosol size gets smaller. So there's smaller particles. And this is a slide, a figure looking at survival characteristics of airborne, just seasonal, run-of-the-mill coronavirus, and looking at the effect of humidity. So under high levels of humidity down here, the virus doesn't survive very long, so not much aerosolization. But when the humidity is reduced, it's considerably, the survival increases considerably, and so does the aerosolization. So that's important as we go towards winter, which is a low humidity type environment. And the other issue is the cold weather. So they also looked at the half-life of the virus under different temperature conditions. So they compared 6 degrees to 20 degrees. And as you can see here, the half-life was much longer, so showing much more survival of the virus in cold temperatures. So low humidity, cold temperatures, that results in more stability. So it's not good news for COVID-19 transmission. And it also makes one wonder about at least the cold, the outbreaks in meat packing plants. I'm sure a lot of you have seen this in the media, where, for example, in Alberta, very large outbreaks, and it's really hard because it certainly could be confounded by the fact that people are working very close together for very long hours. But one wonders, is there an effect of the cold temperature in increasing the transmission? So those are bad things. I guess one good thing is that, at least experimentally, the virus is rapidly inactivated by simulated sunlight. So this was a study from earlier this year where the investigators looked at darkness and then looked at a medium amount of light. This is the result was the viral aerosol concentration decreasing more when they increased the amount of sunlight. So being outside when sunlight is a good thing. Now, in terms of general seasonality, this is a slide just looking at historic rates of seasonal coronavirus in people who had upper respiratory illness. So you could see that it's very consistent, that it occurs season after season. And that's something we will probably see in the future with SARS-CoV-2. There have been a number of predictions. This is one from a group of Harvard that said, well, if we look at the duration of immunity, and if we say that the duration of immunity is going to be very short-lived, well then, it'll look like the flu. There'll be peaks every season. If, on the other hand, the immunity is really long, so that we're protected for over 100 weeks, there might not be much COVID around. And if it's in between, maybe every second year there'll be a blip. So again, this remains to be seen. We really don't know how long after someone is infected or how long after someone gets a vaccine, an effective vaccine, will they be protected for. Now, in terms of the effect of temperature and humidity with respect to potential spread of coronavirus, this is sort of an interesting study where they looked at 50 cities and they looked at 8 where there was a lot of COVID transmission, and they compared that to the 42 where there was nothing. And they found an interesting, they had an interesting finding that most of the cities where COVID was being spread had mean temperatures between 5 and 11 degrees Celsius, and they were in relatively low humidity areas. Okay, moving on to another topic, which is relatively controversial as well, just like the transmission, is how can we prevent transmission of COVID-19? And I'm just going to focus on personal interventions, physical distancing, face masks, and a vaccine. So as I've been alluding to, there's sort of two solitudes of opinion, where the aerosol biologists tend to place the greatest weight on experimental data, the things that they control very, very carefully, versus the clinical scientists and epidemiologists who rely on epidemiologic data and evidence-based medicine. And this is one example. This is a study done by a group of engineers in Hong Kong who basically did modeling and try to find out where large droplets dominate versus aerosol domination, depending on the distance between two people. And they said, look, after half a meter, it's all aerosol. So it's all aerosol domination. And that's certainly in contrast to the evidence, let's say the more epidemiologic evidence. This is a study by Derek Chu and Holger Schoenemann, that did a systematic review, and they point out here, if you look at the absolute risks of infection, well, it certainly goes down if you're separated at a distance between 1 and 2 meters. It doesn't mean, you don't have to necessarily be 6 meters away or 7 meters away. So it just highlights sometimes the difference between the different ways of looking at risk factors. And I'll also, what you want to use to inform policy. And here, this is another very controversial area, which is what is the appropriate type of face mask healthcare workers should use. On the one side, there's these N95 respirators, which are, they're super well designed. They filter particles to 0.3 microns. They have to be fit tested. They're leak proof, et cetera, versus the plain old surgical mask that the filtration properties aren't very well known, but they tend to be used for droplet. And then, so there's a sort of a controversy where the WHO is recommending the surgical mask. So is Canada, CDC, and European CDC say no, it should be the N95 respirator. And this is for relatively low risk activities. My view personally is that we should be, if we do a vaccine study, we don't rely on pre-experimental data. We do randomized trials. We do phase 1, phase 2, phase 3. And to me, it's the same thing. If you're looking at medical devices to protect healthcare workers, you still want to be looking at that higher level of evidence. So Derek Chu in his study looked at the association of face mask use for sort of COVID-19 type viruses like SARS and MERS, and found about a 70% risk reduction no matter what you wear. And he also looked at the N95 respirators versus surgical masks, but these weren't head-to-head comparisons. He found a bit of more of a benefit with the N95 respirator, but there were a lot of problems with some of these studies. Some of these were healthcare worker studies, etc. Jessica Bartosco, who's a PhD student here at McMaster, did a nice systematic review and found, well, if you look at the randomized controlled trials, there's actually no difference between, or very little difference between the two. And in fact, we did a randomized controlled trial a number of years ago looking for flu, looking at surgical mask or N95 respirators to protect nurses against the flu. And we just happened to look at coronavirus. Incidentally, there was no difference in attack rates or infection rates, whether they were wearing the N95 or the surgical mask for influenza. But we looked at coronavirus, and there we found the infection rate was a little higher in those that were wearing the N95 respirator versus the surgical mask, but it wasn't statistically significant. So the bottom line is we need to do a randomized controlled trial, which is what we're doing now in Alberta, Quebec, and in Ontario, and in other countries, to randomize healthcare workers when they're looking after COVID patients to either the medical mask or the N95 respirator, and just to see whether our hypothesis is the medical mask will, just like the flu, will be just as good as the N95 respirator. Now, it's very, another controversy, of course, well, for some people it's a controversy, it's the face mask use in the community. And if you look at the randomized controlled trials, there are no randomized controlled trials for COVID-19. These are for flu. If you pool them all together, they don't show a dramatic effect, but they show, depending on the studies, 10 to 15% effect. So I would say at a population level, it's important. So there is randomized controlled trial evidence that in addition to physical distancing, it's important to use face masks in the community to protect oneself against COVID-19. So lastly is the vaccine. And so this is, again, it's an emerging game. There's no vaccine yet that's been shown in a phase 3 study to be effective. What's sort of new is that the platform, there's these so-called pathogen agnostic platforms, which means that these are platforms that, it could be COVID-19 or it could be, another virus that's used to protect people. So as you recall, I showed you the spike protein. So basically, if the gene that codes for this protein is isolated and it's put into another virus like an adenovirus, and then it's injected into people, it'll cause an immune response. So these types of vaccines are being tested right now. There's a type which is a subunit vaccine. It's just basically you take the gene, you make the spike protein, and then you get a lot of copies of the free-floating spike protein. And you have these nucleic acid vaccines, which are just bits of either RNA or DNA that are taken up by the cell, and they produce an immune response. So this is the landscape of COVID-19 candidate vaccines, and these are just, there's over 200 in different phases. These are the phase 3. So phase 1 is sort of first in human studies, just to show safety. Phase 2 is to look at the immune response, and then phase 3 is these large studies, which are over 10,000 people, up to 40,000 people. And you could see that a lot of these sort of pathogen agnostic platforms are being used with non-replicating viral vectors or RNA viruses. So unfortunately in Canada, we really don't have the biomanufacturing capability to scale up and make vaccines. So the government has had to negotiate procurement deals with US-based vaccine companies. One potential side effect of a SARS-CoV-2 vaccine is the so-called Th2 response. So this is, without going to a lot of details, a response where somebody gets the vaccine, and although it might actually be effective in protecting against COVID, it causes a collection of these eosinophils. Now, the good thing about this is most of the phase 1, or all of the phase 1 studies, at least that have been published, have been shown really a Th1 response and not a Th2 response. So hopefully we're not going to see these types of adverse events in these phase 3 vaccine trials. And as most of you are probably aware or might have heard, the timelines for the vaccines have been super accelerated. Normally there's a phase 1, which is again a safety thing. Then it goes to phase 2 and a phase 3, and usually they're sequential. Now they're all overlapped, and in fact, the manufacturers have to start producing doses of the vaccine before they even know whether in a phase 3 trial it will be effective. So it's something that usually takes over 10 years that's been stretched out in a year and a half or 2 years or even less. So I'll just summarize that transmission appears to be predominantly short distance, but it's important to remember that there are multiple routes of transmission that are possible. The cold weather and low humidity appear to stabilize the virus, and physical distancing and face masks currently, in the absence of a vaccine, are really the best prevention strategies. Thank you very much.
Thanks, Dr. Loeb, for your insight and your expertise. Very, very useful knowledge on a range of different topics. Now I would like to open this lecture for question answers. So please type your question in the QA box that you see on the side. Just for going forward, you were talking about the vaccine timelines. There has been quite a controversial reports coming. In fact, the WHO announced that it may be by the end of year, and few days ago we heard from Government of Canada, some representative, it may take several months. So what may be the timeline, optimistic, realistic timeline going forward?
Well, you mean I'll have the timeline for a vaccine to be made available to people? Is that
yes.
The question?
Yes.
Yeah. Again, without knowing the results of the phase 3 trial, it's impossible to say. Again, we're at the mercy of these sort of reports. So I really don't know. But there's one thing. Even if let's say there's a vaccine that's effective, let's say this year, November, December, there's results of a phase 3. What does that mean to us as Canadians? Well, first of all, there has to be distribution. It would be distributed to people in the United States. So one thing that's clear to me is that we would not be getting the vaccine before the population of the United States, or at least I can't say it's clear to me, it's relatively clear to me that we would not be at the top of the line, that the countries that are manufacturing the vaccine will have they'll get it first. And there are a number of countries that have said this, even India, for example, has said, well, if they're manufacturing a particular type of vaccine, they wanted to have it distributed to their population first. Even France has said, well, if we're going to make a vaccine, we want it to go to our population first. And so I think that if there's procurement deals to the United States, I'd be shocked if Canadians go to the front of the line. So I think it's going to be a while before we get a vaccine.
Okay. So on the related aspects, so there's a flu season already starting, and the flu shot has already arrived. So should people go for the flu shots? Would there be added benefit? Because some may argue that flu shot may also enhance your immune system, and then enhanced immune system may be not good. So any clarification on this?
There's no question that getting the flu shot would be good, because first of all, if there's flu circulating and you get infected, that's bad, and the more people who get infected is bad. And if you get infected, you could transmit it to someone who is a high risk of complications, which is bad. And it's sort of doubly bad now because there's the diagnostic uncertainty, right? Because the flu symptoms and the COVID symptoms are basically virtually the same. So that if people are getting more symptoms that are the flu, and that we have to use up more diagnostic tests to figure out, is this COVID or this is flu, it just complicates things. That's, two or three reasons right there. Other reasons are co-infection with influenza and COVID-19. So those people who are at risk of complications of one often are at risk of complications of the other. So, you don't want to be in a situation where you're getting a double type of infection. So now there's there's a theoretical benefit of, whether if you get a flu shot, is there sort of innate immune response that will protect you from COVID as well? That's very hypothetical. Personally, I would be surprised, but I think there's enough good reasons to get the flu shot that are just standalone.
Okay, that's good. So the clear message to the audience is get your flu shot. So
yes, absolutely.
That's good. And then there were a number of questions that has been asked for the children. So the number one question is that should the children be in the school? And if they're in the school, then there is schools are open, universities are closed. So what is your message to the parents for the children?
Okay, well, that's another one of these controversies, right? Where you have people looking at the same data, and I'll preface my argument this way. Okay, so I think that schools, if there's a school that's in a jurisdiction that's having high rates of COVID transmission, really high rates, to me, it's likely that the school might be an amplifier of the COVID situation there. So that's not good. If it's if there's no COVID activity whatsoever, then the school would be, it'd be a lot safer. And of course, there's a lot of childhood developmental reasons for children to be with other children in schools. You see, so it's certainly a complex situation, and you see people arguing back and forth for both. But so I would say, overall, personally, I think it's good for children to be in school. It would not be a good idea if there's a crazy outbreak in the vicinity, because then I think that would just increase the transmission. So, it's sort of maybe a sort of it depends type of answer.
Okay. So in a lot of common circumstances, when the children go to the school, so then there's a lot of they're sick. So there's a temperature, there's a running nose. So all these basic things come, the sore throat. So at what point a parent should be worried for the testing and should be going for the testing and differentiate between the common flu versus the coronavirus?
Well, yeah, so, those again, they're good questions. So what we know so far is that for children, the complications of COVID are they're not as dramatic as for adults. The testing recommendations to me have been a little bit sort of confusing. So I just want to make sure that I understand the question. At what point? So I think from the clinical situation, when you're looking at these respiratory viruses, it'll be virtually impossible to say, is it one or is it the other? I like to say that you should be obeying what the public health authorities are telling you, but I understand that sometimes there is mixed messaging, and it's difficult if you're a parent. You're hearing all these different things. I would be a bit wary of going to Twitter and finding all sorts of, because you can find anything on Twitter, and it's so polarized that it's very, very difficult. Now, and the other thing about the testing is that right now, it also depends where you are. Okay, that's another point, because in some jurisdictions right now, testing is being prioritized for people who are really ill, for inpatients, etc. And that's what we're doing, because we don't have the testing capacity. So I don't think at this point in time it's a good idea to bring every child to get tested who has a runny nose, because they're not going to be tested, and if they do get tested, the turnaround time will be 5 or 6 days. So it's a difficult one, and for some of these questions, there's no easy answers. I think for the benefit of children, I think it is good for them to be in school, though. That's my own view.
Okay, that's good. And then so we are already seeing the start of the numbers are going up. So what we expect, how worst we may go, and then in January we hit -30, in some areas in Canada -15. So really, really cold temperature. Would they help in some ways, or how? Because the temperature range you gave was like a 5 or 10,
Yeah, I don't know that the extreme cold weather is going to be more helpful. I'll have to be very honest with you. I think cold weather will lead to more people indoors, and indoors relates to transmission. So I think people have to be really worried that, if you're outdoors in sunlight, you're relatively safe. I think there is a concern indoors, and whether, again, whether we go about the droplet versus aerosol transmission debate, I think it doesn't matter. I think people have to be really careful, really cautious indoors to physically distance themselves and to use a mask. And I think things have the potential to get worse. I myself, obviously, I don't know how bad things are going to get. It's very difficult to say. I certainly hope it doesn't go to an extreme. To a large extent, I think it'll depend on what the public health interventions occur. I think in an ideal world, there would be very detailed surveillance in your own region. For example, let's say around Hamilton or whatever, and there would be a system of taking swabs, of sentinel markers, maybe a bit in schools and nursing homes, so that we have a very good, firm understanding of what's happening. Okay, in an ideal world, and every region does that, and maybe it brings that centrally, and then there are decisions made based on those data, very data driven. But right now, that's not what's happening, right? There's not the capability, there's not the resources, there's not enough testing going on. There doesn't seem to be, a lot of centralized information. Maybe there is, but I might not be aware of it. But there's certainly not a really sort of systematic surveillance going on that will help to answer these questions about schools, about, how bad things are going to get, etc.
Okay. So the one other question that I would like to ask with respect to the climate change debate. So we already have strong data on the vector-borne disease. So 1 million people die from the vector-borne diseases and their range of diseases. And then the climate change will put in, there's evidence that they will be increasing. And now, are we looking for a double impact? Now, in addition, on top, there's a COVID. We already seeing increase in vector-borne diseases. So what would be your message, or what cautious, or what you will suggest going forward on this point?
Well, I think the climate change and, with increased temperatures and vector-borne disease, that in itself is something that is, is a real concern. And I think in of itself is something that needs to change, in terms of any interventions. In terms of impact on COVID, I can't say. All I can say is, well, COVID makes anything worse, but we don't want to be in a situation where years from now there's, dengue, there's malaria, there's all sorts of vector-borne diseases. So I think, COVID aside, there's enough evidence to say that climate is making a difference, and it's a concern for infectious diseases.
Okay, thanks a lot. Thank you very much. So I would like to bring our talk to a close, and I would like to thank Dr. Mark Loeb and Dr. Maureen McDonald to be with us tonight. So we really appreciate their time. And I would also like to thank our partners, Global Water Futures Program and United Nations University. And a special thanks to our team members who has worked behind the scene to make this event a success. So they can wave you, they can unmute their cameras. So thank you very much from our team. Please continue to be responsible, stay safe, and practice social distances and wear your mask. So thank you very much. Good night.